So, welcome back to the lecture series in animal cell culture. So, in the last class, we kind of walked through a virtual lab, which I wish most of you develop someday. So, what are the points which has to be kind of kept in mind in the whole layout and the budget constraints what you face? So, now I will devote a little time on some of the aspects or some of the facilities which are kind of advanced level facilities, which most of the time are not being taught in a cell culture lab, but some of you may need it to think in that way too. So, we have already put the layout. Now, if you have to have an addendum of certain things, why I told you leave a lot of space. So, that is precisely is the point. So, today while we are moving on to our week 3 lecture 3, we will talk about those W3 L3. We will talk about those specialized situation, specialized facilities within a cell culture lab if you wanted to develop. So, one of the thing which you may need, suppose you need it if you are working on space biology and you needed a cell culture facility. So, you have a cell culture facility, so you have to curve out a niche of certain spots to have certain specific experiment. And one of the critical experiment what is being done in space biology is where you wanted to simulate the growth of cells in an in vitro culture using a zero gravity simulator, which the most simplest of all is called a clinostat. Clinostat. So, this is a very interesting thing which kind of it's a setup which rotates like this at a particular RPM, and you have your sample kind of kept somewhere out here. So, the cells experience a change in the gravity like this. So, the gravity value changes. These kind of devices, Panasonic in Japan, they do make it, and if you have quality engineers around you, they can develop it for you. There are designs which are available online to make these kind of devices, and their whole idea is to simulate zero gravity, or zero gravity is tough really to simulate. It's basically a microgravity situation, how the cells grows in that kind of situation. So, you have a cell growing dish out here from where there won't be anything will spill over. So, the inside it's sealed properly with of course proper gaseous exchange, and it is moving in this clinostat. Okay. So, if you have to have a facility like that within your system, so you have to go back and see where you really can accommodate it. So, you have to have these spots which are left somewhere out here, say for example. So, that is why I told you that leave space, you do not know where you may be needing things. Okay. Or say for example, you may need it to, say for example, you have a space biology setup out here. This is one specialized setup you are adding into it. Or say for example, you wanted to create certain situation of unusual extreme environment, unusual extreme environment situation. Something like you want to simulate conditions of poles, so North Pole, South Pole. So, you needed incubators of that kind where you can simulate those conditions. So, you needed to keep them at separate spots. So, you never know when you are planning a lab, you really do not know where you are going to land up with. But you have to have a provision that, if I land up with it, I'll be working on it. Then there are several labs across the world who are currently working in the area of integrating cell on microstructures, which you must have heard something called biomems. M stands for micro, E stands for electro, the second M is mechanical, S stands for system. Biological, micro, electromechanical systems. These are essentially microstructures. So, and we'll be coming later about it. There is another buzzword in the market which is called lab on a chip, which is nothing but a small setup where you are growing the cells, which is self-contained, so you have supply of medium and everything within it. We'll come later. At this point, just listen to me and think over it, but we'll come systematically on this topic. Now, for these kind of work, you may need it to carve out a space. Either you do it somewhere out here in the outer room, where, so you have the autoclave here, but then you have a space out there. You can use this, and not only that, there are these kind of setup added up with electrical recording setups. So many of the labs who work on these kind of electrical recording systems, you have microelectrode arrays, field effect transistors you work on. So you needed to have those kind of assemblies already inbuilt into it. Or say, for example, 4th year or 5th year down the line, or in next 10 years you want to develop, then you have to have the provision already there, how you want to develop it. You cannot make this call at the last minute that, okay, now I want this. That won't work. So you have to, because the reason why I am adding up to this, people always think we'll have another room where we're going to do it. But the thing is that your biological samples are there. So you might as well think little louder ahead of your time and have the things then and there, so that you minimize any kind of contamination. Because see, your major problem will be contamination in any of these cultures. Following the aseptic techniques, you must have seen all throughout, I'm repeatedly kind of hinting upon it, not really telling that this is you should do, this is you should do, but I'm kind of making you feel, as we are developing this virtual lab, how it will look like, that have the sticky mats, have the wind or the current, have the room continuously circulating it, how you are going to work on the laminar flow hood, where you should put the laminar flow hood, where you can have minimize the outside current flow, where you can have sliding doors. So these are small bits and pieces of informations which will help you to follow the aseptic techniques. So if you have plans to explore in the area of microelectromechanical systems and all those kind of things, you might as well start thinking in the beginning instead of thinking after you make up the lab. Or you have a colleague who is part of this team who wants to work on electrodes and integrating cells on electrodes. So you have to provide that small niche within this assembly where that XYZ individual can come and do certain things. So think in advance. Or there are people who work on kind of interaction of toxins with the cells. So they may need a very separate hood where they can play out with their toxic material and see their effect on the cells. So you have to be careful, because this individual is working on something else, and you have to may prefer to have a separate incubator for those kind of cultures, because you do not know, because you are testing something very toxic, it may mix up with something, right? So those are mostly people who work on some kind of industrial related problem, or some kind of other toxic material derived from insect, fungus or something. So you have to think several times. And another thing, try to avoid a close proximity of a microbial facility along with a mammalian cell culture. Try to avoid it. You may have a situation you have no other go, they will be close by, but try to avoid it as much as you can. That let there be a distance, or if you have no other go, that you have a microbial facility adjacent to your cell culture, you have to ensure that all the aseptic techniques are being followed, and you have to be ultra careful at every level that microbial contamination should not contaminate your mammalian cell culture facility or animal cell culture facility, or whatever cell culture you are following. Keep that in mind. These things no one will tell you, but then if you have no other go and you have to really have a lab cell culture facility adjacent to a microbial facility, then your only option is that you just implement strict code of conduct so that contaminants from one site does not spill over into the other one. So this is another thing which you have to be very careful. So you see, as of now we have discussed about some of the specific areas. This area may go a long way. So I have only talked to you about, say, suppose you have a facility of working on space systems, space biology, or you are creating a system of extreme environment, so you have to have separate incubators, or you are working on something like micro electromechanical systems and electrical recordings. So you may love to have all these facilities in and around you, and these are the kind of things where these are helpful for. We'll be coming later into this about microfluidic setup. There are people who are working on microfluidics, there are people who works on reactor designs or microreactors. Okay, they are working who are working on gas exchange assemblies. There are people who works on lithography. So these all fall under the basic purview of micro or biomems. So keep all these in mind when you are setting up the facility, how you are going to do it. Shall we split? So there you have multiple ways. Suppose I get a facility like this. I split up into 4 different chambers for different purpose. So from one corner you are entering and then you rearrange it. So I have a facility for X, Y, Z likewise. So you have multiple ways, or the one which we kind of in virtual system develop, like 2 part 1, part 2. You may have part 1, part 2, part 3, part 4. It all depends very. So these kind of drawings you have to do in your basic manual to visualize what you are asking for. Whatever you are going to asking for, you are going to get that. But unless you do your homework right, you will land up in trouble. And what are the points what you have to keep in mind? What is your, again rehashing it, what is the number of user? This is very important. Second, what are their different areas of interest? So that will decide what all resources you will be needing, right? and all these things are a function of the kind of budget you're looking forward to. And most importantly is, what are the milestones? In terms of milestone, I meant, like, suppose today I am starting 1 year, 2 year, 1, 2, 3, 4, 5, 6, okay, likewise. So within this year we'll achieve this, with our next set of funds we'll do this, next set of funds do this. So that demands that you chalk out the space reasonably well. So do not unnecessarily cover up the space with bulky stuff which you know you have to remove. I have seen people, oh no, I'm covering the space, it should look all filled, which is a foolish mentality. Do not do that. Think over it, that if I have a plan for next 10 years down the line, or I have a plan for next 20 years down the line, why I should unnecessarily cover up spaces? Let it be there, let it remain slightly empty, no problem. Another thing what I did not mention here, while you are planning this setup, on the walls of these sites you will have storage of materials. But now if you want to do it on the walls, then you have to keep in mind, then you have to have a dedicated ladder for your lab, which is purely dedicated. You have to keep the ladder somewhere, or nowadays we have all these kind of foldable ladder or something. You have to have a foldable ladder because you have to pull down the stuff from the racks on the sides. Okay, so keep these small tips in mind. It will be very helpful once you make the lab. Now comes what kind of lighting you are looking forward to. It is a good idea to have a facility with really bright, good quality lighting, and every day ensure the whosoever is coming in and out, first of all they should be very careful about their, point 1 on the aseptic techniques, I'll tell you about their personal hygiene. This is must, the must. Your personal hygiene, there is no compromise on it. If you are not well, it is absolutely advisable that do not come to lab. Second, so personal hygiene. Personal health. Next, it is absolutely make it a mandatory point to have the hair caps, so that if there is a hair fall or something, it is not going to contaminate the system. Have the mask, even if you have a mild cough or anything, it is not going to contaminate the system. Have your lab coats cleaned on a regular basis. Ensure you wash your hands all the way up here before you kind of wipe it with alcohol, slight alcohol, just wipe it, and before you put on the gloves. As long as you are following some of these very basic fundamental rules, and when you are inside the lab, be dedicated for that period of time. Do not get I'm doing this, I'm talking, I'm doing, like, do not do those effects, because whenever you have to talk, you have to remove your mask. So I have nothing against the talking, and just remove your mask, and you spread several things all over the place. So, and especially this holds true when you are on the laminar flow hood, when you are working on the laminar flow hood. So on the laminar flow hood front, The very first criteria is wipe the surface and the side and the back after you finish the work with alcohol. Before you start the work, do another wipe. Okay? And once you finish the work overnight, keep the UV on at least for 2 to 3 hours before you start the work, or you can even leave the fan on. There is no harm, but if you keep the fan on, then you have to keep the front opening also open so that the air continuously flows out. And every week or weekend, schedule your lab in such a way that this laminar flow hood is being inspected and cleaned. Then comes your incubator. While you're opening the incubator, be very, very ultra careful about your aseptic techniques in terms of the incubator. Incubator is your lifeline in a lab because that's where all your cells are. So you should have already planned a designated trays or shelf for individuals. Do not fight over it. Let there be designated things. And before you put your samples and everything, just with a mild alcohol, wipe the stuff, keep it there, and ensure that rack is being wiped out at least on, if not regular basis, at least once in a week. Second thing, please ensure that there is always a tray underneath the incubator, that water is being changed, and there are certain antimicrobial detergents which are present. You please add in that. Ensure that. Very, very essential. And suppose you are placing your stuff inside the incubator. First of all, minimize the opening and closing of the incubator. You have to plan it. And suppose there is spillover, immediately wipe it out, pull out the tray, wipe it out inside the laminar flow hood, and then put it back. Do not neglect. Another important thing what I have observed over the years is people forget to keep track of, if it is a CO2 incubator, of the amount of CO2 in the incubator. So some people, I know once I did it, they have a sensor which will say if the CO2 level goes down below a point. Of course, the incubator has sensors. We had additional sensors what we added. So have, say for example, you have one incubator here, right? And of course, for that one incubator, you have a cylinder adjacent to it. So you have another incubator stacked on top of it. You have, say, 2 cylinders hooked up here, like this. I'll recommend you have 2 more standby behind them for the rainy days, because your friends will forget that these are about to end, and then you are hovering, running around, what to do. Don't allow that to happen. So have these backups ready. The day you fix this cylinders, maintain a logbook, and on top of that put a sticker telling that fixed on, say, day, month, year. So you have a fairly good idea that whether it will last a month or 2 months or whatever. So you make it a scheduling. It's a very scheduling problem which most of the people forget. You have to do a lot of scheduling with your colleagues that, okay, next change. So as soon as these 2 are over, we'll have to immediately send them for refill, and in the meantime this will come. And from, you remember on the outside room I told you that outer room you'll have the backups ready. Those backups from that room will come and replace these 2 green ones, and these 2 green ones will take the place of the blue ones, and these 2 blue ones will go for refilling. So you have to have a log register keeping track of it, and you have dedicated members of the lab will ensure that your lab never runs out of it. This is how much soever boring I may sound. Trust me, I have seen people, I have seen labs really in a crisis that nobody bothered, and one fine weekend, everything went for a toss. Second thing, which is very important, is you may opt for a uninterrupted power supply somewhere into this setup. This UPS facility is very important because of the obvious reason, because you are running all these kind of incubators out there, and these incubators, if the light goes off, they will go off, and that's something you cannot afford. So you have to have them on dedicated facility or dedicated line, and same way you want the refrigerator to be on the dedicated track. These are something which you have to take precautions well in advance. Laminar fluid you can avoid, fine, there is no light or no power, you can get around. But in India, this is not only India, I mean across the world, this is a problem. There may be sudden power surge or sudden power drop. How you get around it? So these are some of the points what you have to be really cautious in terms of safety protocols, in terms of aseptic techniques, in terms of backup plans and everything. So I'll close in here and we'll continue this discussion on other different aspects of it, but please be careful and do your designing right. Thank you.